Welcome to podcast 1D.
1D.
Water, pH, acids, and bases. The foundation of biochemistry, part of our BiochemSerye,
Yeah.
Learning on the Go series.
Learning on the go.
Today, we're diving into some seriously fundamental concepts that, as biochemistry professors, we find ourselves explaining over and over to our students.
Yeah.
It's the stuff that makes biochemistry tick.
It's true.
So let's start with the molecule that makes life as we know it possible.
Okay.
Water.
Right.
You'd think something with a formula as simple as H2O wouldn't be that complicated.
Right.
But water is truly extraordinary.
Oh, it is. It really is. And even in our lectures, I find myself, even with seasoned researchers, sometimes we have to revisit these core ideas because they're deceptively simple, but they underpin everything in biological systems.
Absolutely. So let's start with comparing water to methane,
Okay.
CH4.
Right.
They're roughly the same size and mass, but their properties are drastically different. Methane's a gas at room temperature, while water, of course, is a liquid.
Right.
And water boils at, get this, a whopping 250 degrees Celsius higher than methane.
Huge difference, yeah.
Okay, that is a massive difference.
Yeah.
What is the secret behind water's superpowers?
Well, it all comes down to, I think, a favorite topic of mine.
Okay.
Hydrogen bonds.
Yeah.
You see, the way that those two hydrogen atoms are bonded to the oxygen in water creates a dipole, meaning that it's like a tiny magnet.
Oh.
One side of the molecule has a slight negative charge and the other a slight positive charge.
So these water molecules are all attracted to each other.
Exactly.
Like little magnets.
Little magnets clinging together, yeah.
Clinging together.
And those hydrogen bonds are surprisingly strong.
Yeah.
They're the reason that water is a liquid at the temperatures that support life on Earth.
Right.
Imagine if all the water on our planet was gaseous, no oceans, no lakes, no us.
No biology class field trips to the beach.
No, exactly.
Okay, so hydrogen bonds are a big deal. But I'm a little confused.
Okay.
You said water molecules are attracted to each other.
Yeah.
Yet ice, which is just water in a solid form, floats.
Right.
Shouldn't it be denser
Yeah.
than liquid water if the molecules are more tightly packed?
That's a great question, and it trips up a lot of students.
Yeah.
When water freezes, those hydrogen bonds lock the molecules into a rigid hexagonal lattice structure.
Okay.
And believe it or not, this arrangement actually spaces the molecules further apart than they are in liquid water.
So they're further apart in solid ice.
Yes, they are.
Mind-blowing.
It is, isn't it?
Yeah.
And this little quirk of water has enormous implications because ice floats, lakes and oceans freeze from the top down.
Right.
Allowing aquatic life to survive in the liquid water below.
Right.
If ice were denser, everything would freeze solid from the bottom up.
Oh, wow.
And life as we know it wouldn't exist.
It's amazing how such a seemingly simple property has such profound consequences.
It is, isn't it?
Yeah. It makes you wonder if life could even evolve on a planet where water behaved differently.
It's a fascinating thought, isn't it?
Totally.
So, moving on to another incredible property of water is its ability to dissolve things.
Right.
We call it the universal solvent for a reason.
It dissolves everything.
Pretty much.
From sugar to salt.
Yeah.
It all seems to disappear in water.
Yeah.
What's going on there at the molecular level?
So it's those dipoles again.
Okay.
Water molecules with their positive and negative ends can surround charged ions, forming what we call hydration shells.
Okay.
They essentially neutralize the charges of the ions, allowing them to dissolve.
So when I add salt to my pasta water,
Yes.
those little water dipoles are surrounding the sodium and chloride ions and pulling them apart.
That is exactly what is happening.
I'm witnessing hydration shells.
You are witnessing hydration shells in action every day.
In action every day in my kitchen.
That's right.
That's pretty cool.
Yeah.
So it's not just salts that water dissolves.
No, it's not.
It also dissolves things like sugar. Yeah.
Absolutely.
Right.
Water can dissolve molecules that have polar groups.
Okay.
Those containing oxygen or nitrogen, for example. And again, hydrogen bonding is a key player here.
Right.
The more polar a molecule is, the more water-loving or hydrophilic it is.
So something like glucose, with all those hydroxyl groups,
Glucose, yep.
is very happy to dissolve in water.
Very happy to dissolve in water. Sugars are very hydrophilic.
Okay, on the flip side, we have
On the flip side,
hydrophobic
or water-fearing
substances.
Water-fearing substances.
And these are primarily composed of hydrocarbons.
Primarily hydrocarbons, like fats and oils, yes.
Which brings us to the classic example. oil and water don't mix.
Oil and water don't mix.
But is it really because they repel each other, as some people say?
It's not so much repulsion as it is a matter of energy.
Okay.
When nonpolar molecules like oils are introduced to water, the water molecules actually start forming these low entropy cage-like structures.
Low entropy, I'm going to need you to break that down for me.
Okay, so essentially what that means is the water molecules become more ordered. And that's not energetically favorable.
Okay.
So the system will naturally try to separate to avoid this.
So it's not that oil and water hate each other.
Yeah.
They just can't hang out without making things awkward for the water molecules.
You got it.
Okay.
And this explains why, if you shake oil and water together, you get all these tiny oil droplets.
Yeah.
But they quickly merge back into bigger drops.
Right.
It's all about minimizing the surface area of contact between the two.
So we've got hydrophilic molecules that love water.
Love water.
And we've got hydrophobic molecules that avoid it.
Right.
But what about those molecules that seem to have a foot in both camps?
Yes.
Like soap.
Yeah.
You know?
Ah, yeah.
It seems to do both.
So those are called amphipathic molecules.
Amphipathic?
The drama queens of the molecular world.
No?
They have both polar and nonpolar regions.
So, like, they can't decide whether they want to be in the water or out of the water.
That's a good way to put it, yeah.
Okay.
Think of a soap bubble.
Yeah.
You've got a thin film of water sandwiched between two layers of soap molecules.
Right.
The soap molecules arrange themselves so that their hydrophilic heads are facing the water.
Oh, okay.
While their hydrophobic tails are tucked away inside, shielded from the water.
So soap bubbles are essentially amphipathic molecules.
Yes.
Putting on a physics show.
I like that, yeah.
Okay.
And this type of arrangement is crucial for life in our bodies. Phospholipids, which are amphipathic, form the basis of cell membranes.
Wait, so the very walls of our cells are built on this?
They are.
Can't live with it, can't live without it.
That's right.
Relationship with water.
That's right.
That's incredible.
From soap bubbles to cell membranes.
Yeah.
Amphipathic molecules are essential players.
This is blowing my mind. Okay.
Right.
Let's shift gears now.
Okay.
And talk about another fundamental duo in biochemistry. Acids and bases.
Acids and bases are it.
I have to admit, even after all these years of teaching, I still find myself reviewing those definitions.
Me too. It's easy to, get lost in the complexities and forget the basics.
Right, right.
So let's get back to the core concepts.
Okay.
Acids are proton donors.
Okay.
They give away those positively charged hydrogen ions, or H+, and bases. Well, they're the proton acceptors.
Right.
Happy to take on an extra H+.
I'm with you so far.
Okay. Yeah.
But you know what always tripped me up?
What's that?
The fact that water can act as both an acid and a base.
Ah, yes.
Yeah.
Water's amphoteric nature. It's like the ultimate biochemical chameleon.
Okay.
Think of it like this.
Fine.
When a strong acid, like hydrochloric acid, HCl, is added to water, it eagerly donates its proton to a water molecule, creating a chloride ion, ClO, and a hydronium ion, H3O+.
So in that case, water acted as a base.
Yes.
Accepting the proton from the hydrochloric acid.
Exactly.
Okay.
Now let's look at ammonia, NH3.
Okay.
It's a base.
Right.
When you add ammonia to water, it snatches a proton from a water molecule, forming an ammonium ion, NH4+, and a hydroxide ion, OH.
Okay.
In this scenario, water played the role of the acid, donating the proton.
So water can be both a proton giver and a proton taker.
It can.
Depending on who it's hanging out with.
That's right.
That's pretty versatile.
It is. It is. And this duality makes water a critical player in countless biochemical reactions.
Yeah.
It leads us to another important concept. Every acid has a conjugate base, and every base has a conjugate acid. It's a bit like a dance, isn't it?
Yeah. The
acid and base are partners constantly exchanging that proton.
Okay.
And the thing is, the stronger the acid, the weaker its conjugate base.
Right.
And vice versa.
Yeah.
It's a bit like a seesaw. When one side goes up, the other goes down.
I'm starting to see the connection here.
Okay.
And this whole interplay between acids, bases, and their conjugate brings us to another crucial concept, pH. pH is something we hear about all the time.
It is.
From shampoo commercials to gardening tips.
Right, right.
But honestly, do most people really understand what it means?
I'd say probably not. It's one of those terms that gets thrown around a lot, but the deeper meaning can be a bit elusive.
Yeah.
So let's break it down.
Perfect. Let's enlighten our listeners.
So pH is simply a measure of the concentration of hydrogen ion, H plus in a solution. And it's measured on a logarithmic scale. Which means a small change in pH represents a big change in H plus concentration.
Logarithmic scales, always a bit mind-bending.
They can be, but think of it like the Richter scale for earthquakes. A small increase in magnitude on the Richter scale represents a huge jump in the energy released.
I get the analogy. So a lower pH means a higher concentration of H plus ions.
Yes.
And that means?
More acidic solution. Higher pH means a lower H plus concentration, which means a more basic or alkaline solution.
Got it.
All right.
So the pH scale runs from 0 to 14, with 7 being neutral.
Exactly. Pure water at 25 degrees Celsius has a pH of 7, perfectly neutral.
Interesting.
Yeah.
So every time I take a sip of water,
yes,
I'm experiencing chemical neutrality in its purest form?
You are, and it's important to remember that this whole pH concept is incredibly relevant in biology.
Yeah.
Because even slight fluctuations in pH can have a dramatic impact on cellular processes.
Dramatic, really? I mean, water is water, right?
Think about blood pH. It's tightly regulated within a very narrow range.
Yeah.
Between 7.35 and 7.45. Even a tiny deviation from this range can lead to serious health problems.
Well, that really drives home the importance of pH regulation.
It does.
Our bodies are talking pH balancing acts. But how do we manage to keep things so stable?
Right.
Especially with all the metabolic reactions going on constantly.
Well, that's where the wonders of buffers come in.
Okay.
They're the unsung heroes of our internal environment.
Buffers. We touched on them earlier.
We did.
But let's dive deeper.
Let's do it.
Remind me again how they work.
Think of buffers as chemical shock absorbers.
Okay.
They resist changes in pH by soaking up excess H plus ions when things get too acidic, or releasing H plus ions when things get too basic.
Okay, so they're like tiny pH regulators.
Yes.
Constantly patrolling our cells and fluids.
That's a good way to put it.
Keeping everything in check.
And the magic ingredient that makes this possible is a mixture of a weak acid and its conjugate base in roughly equal concentrations.
So we're back to that acid-base duo, always working together.
Always working together.
Right.
Let's take a class. CH3COOH is our weak acid.
Right.
An acetate, CH3COO, is its conjugate base.
Okay, I'm visualizing the acetic acid and acetate molecules on that seesaw again.
Perfect. Now imagine we add a strong acid to this buffer solution. It starts pumping H plus ions into the mix, pushing the pH down.
So the seesaw's tipping toward the acidic side.
It is, but here's where the buffer kicks in. Those extra H plus ions react with the acetate ions, forming more acetic acid. Essentially, the acetate is acting like a sponge, soaking up those excess H plus ions, preventing the pH from plummeting.
So the buffer is shifting the equilibrium to keep the pH stable.
Exactly, and the beautiful thing is it works in both directions. If we add a strong base to the solution, the OH ions will react with the acetic acid, removing H plus ions from the solution and forming more acetate. This prevents the pH from spiking upward.
So it's a dynamic system.
It is.
Constantly adjusting to maintain balance.
Yes.
It's amazing how nature has figured this out.
It really is.
Okay, so we've got this concept of buffers consisting of a weak acid and its conjugate base.
Right.
But earlier you mentioned that buffers work best within a specific pH range.
Yes.
What determines that range?
That's where the pKa of the weak acid comes into play.
Okay.
Remember, the pKa is the pH at which the acid is half dissociated.
Right.
Meaning there are equal concentrations of the acid and its conjugate base.
That's the balancing point on our seesaw.
Exactly. And a buffer is most effective at resisting pH changes within about 1 pH unit above or below its pK.
So if we have a buffer with a pK of 5,
yes,
it'll work best in a pH range of 4 to 6.
You got it.
Okay.
Outside of that range, the buffer's ability to resist pH changes starts to weaken.
Right.
It's like the seesaw is getting too lopsided.
Makes sense.
Yeah.
So when choosing a buffer for a particular experiment or biological system, we need to consider the desired pH range.
Right, exactly.
And choose a buffer with a pK that falls within that sweet spot.
Exactly. It's all about matching the buffer's properties to the specific needs of the system.
Right.
Now, remember earlier when we talked about how even small deviations in blood pH can be dangerous?
Yes.
Well, that's where our body's own buffer systems come in to save the day.
Ah, yes, those pH superheroes.
That's right.
You mentioned there are several different buffer systems operating in our bodies.
Yes.
Can you tell us more about the key players?
Sure.
I think our listeners would be fascinated to learn how their own bodies maintain this delicate pH balance.
Absolutely. Our bodies are like finely tuned orchestras of buffer systems, all working in concert to maintain harmony.
Right.
One of the most important is the bicarbonate buffer system,
Okay.
which is the maestro of blood pH regulation.
So bicarbonate is like the conductor of the blood pH orchestra.
I love that analogy. It works perfectly.
Okay.
It's a remarkably efficient system that involves carbon dioxide, CO2,
yep.
carbonic acid, H2CO3,
okay,
and bicarbonate ions, HCO3.
It sounds like a complex trio.
It is a little bit, let's start with carbon dioxide.
Okay.
It's constantly being produced as a byproduct of cellular metabolism.
Right.
As CO2 enters the bloodstream, it reacts with water to form carbonic acid.
Okay.
And this reaction is sped up by an enzyme called carbonic anhydrase.
Okay.
One of the fastest enzymes in our bodies.
So we have CO2 from our cells.
Yeah.
Water from, well, everywhere.
Everywhere.
And a super speedy enzyme. And voila, carbonic acid.
Carbonic acid.
Okay.
Now remember that carbonic acid is a weak acid.
Right.
So it can dissociate into bicarbonate ions, HCO3,
wow.
and hydrogen ions, H+.
Uh-huh. We've got our weak acid and its conjugate base.
We do.
I knew they'd show up again.
They're the stars of the show.
Okay.
And here's how the buffering action works.
Right.
If the blood pH starts to drop,
yeah.
meaning there's an excess of H+ ions,
yeah.
those extra H+ ions will react with the bicarbonate ions, HCO3,
right.
forming more carbonic acid.
So bicarbonate is mopping up those extra protons to prevent the pH from dipping too low.
Exactly. And the beauty is that the carbonic acid can then be converted back into CO2 and water.
Right.
Which we exhale with every breath.
Wow, so our respiratory system is directly involved in pH regulation.
It is.
It's all connected.
All connected.
Okay.
And conversely, if the blood pH starts to rise, meaning there are too few H+ ions, carbonic acid can dissociate further, releasing more H+ ions to bring the pH back down.
So it's a constant push and pull.
It is.
A dynamic equilibrium working to maintain that narrow pH range.
Yes.
Our bodies are amazing.
They are. They truly are. And this bicarbonate buffer system is incredibly efficient.
Yeah.
It's estimated to handle about 70% of the acid-base regulation in our bodies.
70%. Wow, it's impressive.
It is.
But you said there are other buffer systems at play as well.
Yes.
Our bodies always have backup plans, don't they?
They do. Another important one is the phosphate buffer system, which is particularly important inside cells and in the kidneys.
It involves the dihydrogen phosphate ion, H2PO4, and the hydrogen phosphate ion, HPO42.
Ah, another dynamic duo.
Yes.
A weak acid and its conjugate base.
They're everywhere.
They are everywhere.
They are indeed, and they work in a similar way to the bicarbonate system, absorbing or releasing protons to maintain pH balance.
So we've got these intricate buffer systems operating in our blood.
We do.
Our cells, our kidneys.
Yes.
All working together to keep our internal environment stable.
That's right.
It makes you realize just how much is going on beneath the surface all the time.
It does. It does. And, you know, when you consider how these buffer systems have evolved over millions of years to become so finely tuned, it's really quite remarkable.
Absolutely. It's a testament to the power of natural selection to optimize biological processes.
It is.
But before we get too carried away with evolutionary musings, I think we should take a step back and do a quick review for our listeners, you know?
It's easy to get lost in the details.
It is.
When you're exploring a topic as intricate as biochemistry.
For sure.
So we started our deep dive with the molecule that makes life possible, water.
Right.
We talked about its unique properties, its high boiling point, its ability to dissolve a wide range of substances, and the surprising fact that ice floats. And all of this we discovered is due to those remarkable hydrogen bonds that form between water molecules.
Those hydrogen bonds are the glue that holds so much of biochemistry together.
Yeah.
They influence protein structure, enzyme function, cell membrane fluidity, the list goes on and on.
It's amazing to think that such a simple molecule plays such a multifaceted role in life.
It is.
Then we moved on to the world of acids and bases.
Okay.
We revisited those fundamental definitions.
Yeah.
Acids donate protons, bases accept them.
Right.
And we marveled at water's ability to act as both.
Water's amphoteric nature is truly remarkable.
It is.
It's a constant participant in the proton exchange that drives so many biochemical reactions.
And we explored the concept of pH, a measure of the concentration of hydrogen ions in a solution.
Right.
We saw how crucial it is for biological systems to maintain a stable pH.
Yes.
And how even slight fluctuations can disrupt cellular processes.
That's why buffer systems are so essential.
Right.
They're like the guardians of pH balance, constantly working to keep things within a narrow, optimal range.
And we took a deep dive into the workings of buffers.
We did.
Exploring how they use a dynamic duo,
right.
a weak acid and its conjugate base, to resist changes in pH.
Right.
We even uncovered the secrets of our own body's buffer systems.
Yes.
Like the bicarbonate buffer system in our blood
Yeah.
and the phosphate buffer system in our cells.
That's right. It's a symphony of chemical reactions constantly adjusting and fine-tuning to maintain equilibrium.
Well said. But before we wrap up our deep dive,
I
want to pose a question to our listeners. Something to ponder as they go about their day.
All right.
We've seen how crucial the properties of water are for life as we know it. So my question is this.
Okay.
How might the unique properties of water have shaped the very evolution of life on Earth? What if water behaved differently?
A wonderful question to contemplate.
Yeah.
It reminds us that life didn't just arise in water.
Yeah.
It arose because of water.
Beautifully put.
Thank you.
And with that thought-provoking question, we'll bring this deep dive to a close.
Okay.
We hope you've enjoyed exploring the foundational concepts of water,
Yes.
pH, and acid-base chemistry with us.
It's been a pleasure, as always.
It has been.
These fundamental concepts are essential for anyone who wants to delve deeper into the fascinating world of biochemistry.
Exactly. And remember, biochemistry is all around us.
It is.
It's in the food we eat, the air we breathe,
Yes.
and the very cells that make up our bodies.
Keep those brains buzzing, keep asking questions, and keep exploring the wonders of the molecular world.
Until next time.
Hey.